Okay, hello everybody, I am Abdalhalim and today I will present my work entitled as Development of Magnetic-Based Biological Sensors for the Detection of Brucella species. So before I start, I would share with you my scientific background. I earned my bachelor degree from Jordan University of Science and Technology in the field of genetic engineering. Then I got enrolled to the master program in biotechnology from the Palestine-Polytechnic University. And currently I am a PhD student in RWTH Aachen University in the field of biology. So in my homeland, Palestine, specifically at the southern part of West Bank, the livestock has a high socioeconomic value. People there rely on it as an important source of food and income. And this livestock has a nomadic and semi-nomadic pastoralism in its nature. However, these sectors have several problems or suffers from different problems. One of them is the shortage in the grazing field, and also it has several devastating infectious diseases that affect this sector. So one of these diseases is the commonest zoonotic disease worldwide, which is called Brucellosis. This disease caused by Brucella species that mainly infect the animals and transmitted to humans by the consumption of the unpasteurized milk and its product. And this disease reduces the livestock production and reproduction performance, evident by frequent episodes of abortion, stillbirth, weak calves and lambs, and also reduction in milk yields. On the other side, the infected human shows symptoms like fever, weakness, severe joint pain, and also the treatment takes several weeks and in some cases months to recover. So why we focused on brucellosis as a model of our detection assay? Because this disease has a very high incidence rate in Palestine. So as you can see here, this is the incidence rate per 100,000 population in Germany and Palestine and Hebron. And from this figure, you can imagine the huge differences between these two countries. So the conventional methods used to detect this pathogen are Ross-Bengal, blood culture, and real-time PCR. Ross-Bengal is simple and inexpensive test, but it has a high false positive rate. Blood culture and real-time PCR, they are sensitive, specific, but the blood culture is time-consuming, real-time is expensive, and both cannot be used in the field. So there is a need to develop a point-of-care sensitive, accurate, with low-cost test to detect this pathogen in the field. So why detection in the field is important? As I said, most of animals lives in nomadic or semi-nomadic areas, which is far from the major cities or big cities that have these central labs. It is important in surveillance because collection of the samples requires a huge money budget and efforts to transport these samples from these areas to the central labs. It helps also in control the livestock trading and smuggling on the border. So thus controlling the spread of a brucellosis between the livestock. So one of these technologies that can be used in the field is the technology called the frequency mixing magnetic detection, which is usually based on the detection of superparamagnetic beads. So this is the magnetic assay for our technique, which consists of capture a probe, interact with the target molecule, and this biological interaction can be sensed by the addition of magnetic bead that will bind to the target. So how this magnetic bead will be sensed? So this is the working principle of our technique. The sample consists of magnetic bead well placed inside a measurement head. This measurement head which consists of three coils. One of them is the outer coil, which is used to generate a low frequency field to drive the probe magnetization toward the saturation. And the second coil used to generate a high frequency excitation field to probe the magnetization state of the magnetic beads. So this behavior of high and low frequency leads to generation of new frequencies. Among these frequencies, F1 plus 2F2 shows the highest signal detection that can be used to detect the magnetic beads by the third coil, which is called detection coil. So started from the problem we have in Palestine and the technology developed in Forschungszentrum Centrum Jülich, a collaboration were established between Dr. Ashab and Professor Krause from Polytechnic University and Forschungszentrum to develop a point-of-care system, sensitive system to detect this pathogen in the field. So our principle of detection will be mainly based on the hybridization between a capture probe with its complementary DNA sequences. And this hybridization can be sensed by the addition of the streptavinated magnetic beads. So if our target exists in the sample, the DNA will hybridize to the capture probe and the bead will bind to it, so the test will be positive. However, when our target does not exist in the sample, nothing will bind to the capture probe, so the magnetic bead will not bind and it will be washed out from the filter and the test will be negative. So our project will go through several stages. First, we need to select a sensitive and specific and conserved target. We will design probes complementary to this target. Then we will choose a proper immobilization method. And we will do some optimization on the hybridization regarding to the time and temperature. And we will test the specificity of the probes. And finally, the sensitivity or detection sensitivity of the assay. So after a thorough bioinformatic analysis, we found a very good target for our assay. This target has 81 base pair length, with 7 copies exist in all Brucella genomes and just only unique for Brucella species. And it has a very high identity from 95 to 100%. Also, this target has a very low secondary structure with a good GC content. So we designed 4 probes with 4 different lengths to detect these targets. First, to detect the PCR analog target with 81 base pair length, and the second target with 50 base pair length. So for the immobilization method, we use the EDC reaction chemistry to covalently bind the carboxylated modified probes to the modified NH2 filter to form a stable amide bond. So this polyethylene filter has a pore size with it 20 micrometer, so any unbound probes will not bind to the filter and it will be washed out. Yeah, to study the effect of EDC, the experiments were done on filters or samples with EDC and without EDC. And as you can see here, the sample with EDC, the amplitude was much higher than without EDC, which was about 0.014 volt. However, without EDC, the amplitude was 0.004, which means that the addition of EDC successfully immobilized the probes on our filter. And in order to test the specificity of the probes, we used two different DNA sequences, one complementary to our probes and one non-complementary. or randomly selected target. So as you can see here, in the amplitude for the complementary DNA sequences was much higher than the non-complementary one that gives somehow similar result to without EDC. So from these results, we can make sure that, or we can conclude that our probes were specific just only to our target we designed. And in order to test how much time it takes for the hybridization, 4 different experiments were done using 4 time different periods: 10 minutes, 20 minutes, 30 minutes, and 60 minutes. And as you can see here, the amplitude of the experiment with 10 minutes somehow gave the same like 60 minutes, which means that the hybridization at 10 minutes was enough for our assay. And in order to test the specificity of the magnetic bead binding, 2 magnetic bead types were tested from the same company, and they have the same size, but the difference is that one have the streptavidin shell and one without the streptavidin shell. And you can see here that the one with streptavinated shell gave much higher signal than the plain one. Then we can make sure that our streptavidin beads are specific in binding to our target DNA. So before I conclude, I want to say that what can be applied on a brucellosis can be applied on any other model in any country. So we can use our detection system to detect any microbes, any pathogen in any country, in anywhere. And that's what I will do next time, to do this test inside the field in Palestine. So as a conclusion, the Hebron magnetic reader designed. We selected a good target. The probes were designed for this target. Now our designed probes were specific to its complementary DNA sequences. The binding of magnetic bead to its bioelectronic also is specific. For the future plans, first for the short-term plan, we are planning first to validate the immobilization method for our assay. Then we will do some optimization on the hybridization reaction regarding to the time and temperature, and we will assess the sensitivity of our assay. For long-term plan, we will select a point-of-care DNA and amplification methods. Then we will select a new primer, and then we will test our assay on clinical Brucella DNA samples exist in Palestine. Thank you.